Good evening, everyone. Today is Tuesday, so we have Taxonomy Tuesday, where our NJMA members share their recent finds or finds of interest. In this case, we're going to have a topic today of puffballs, followed by the member finds, because we did have some warm up here, and hopefully people were out looking. So I have a slideshow. If you have anything to share with puffballs or member finds, please put your name in the chat so we'll remember to call you in order when we get to that. I have about 20 slides about puffballs. So let me try to share. Can everybody see my screen?
Yes.
It's covering up the play. Okay. Okay, so one of the reasons I volunteered for the um, puffball topic that people were suggesting was that I did buy the new Bissett book that came out last year about puffballs. I didn't get much chance to use it because we probably only found a couple of puffballs at the end of the season. Uh, just go back here. It's moving on me on. So the book covers quite a lot, but it's a collection of everything. The previous books that we have in our club library are from the 70s and 80s, which are black and white and not as useful as color photographs. As you'll see, the puffballs actually have color, which also will help identify. So the genera that are puffballs are divided. Oh, this thing is covering up too much here. Come from more than one order. So they're called polyphyletic because they evolved multiple times in history. So they come from the agaricus, which is the gill mushrooms, and they also come from the boletes, which are the pored mushrooms. So we'll find, I'm not going to cover the Earthballs, which are the Sclerodermas and the Pisolithus. I'm just going to try to cover these other genera. You'll notice that some of the genera that we've associated with puffballs, like bestseller, seem to have disappeared. Based on DNA studies, some of them have been moved into Lycoperdon. And in fact, this Calvatia has also sometimes been moved into Lycoperdon. It's sort of a moving things back and forth, as well as Bovista. Those things all seem to be in flux right now. So just some basic terminology. Puffballs will often have two layers of this peridium, which is a container that holds the scleba, which is the spore-bearing mass, because gastroids have their spore-bearing mass internal as opposed to coming off the gills or the pores of a mushroom. With the puffball, there may be two or more. There's an outer peridium, which may have scales or spines, and underneath that will be another layer after those scales, fall off and the outside goes. And there's also talk of a mesoperidion, but I don't have an example of which ones would have that middle layer. The one that we're most familiar with had been renamed fairly recently. The genera used to be Lycoperdon, but now it's Apioperdon, periforma. This is one of the ones that grows on wood. And so those are the ones that have a little pore opening, because that's like a Lycoperdon, which have a little pore opening when they're ready to disperse based on rain or touch. I just want to show you they sort of pear-shaped, like other Lycoperdons. Most of the pictures I have in this presentation are from Dave, an our club member. He is also credited in that Bissett Puffball book with having contributed. So his photographs were very useful to him, and that were very useful for us too. Some of these puffballs are saprobes. In this case, they are on hardwoods and conifers for the wood growing one. One of the genera I was not familiar with is Arachnion. album, which was new to me. This one has a granular mass of peridioles, and it's supposedly because it's growing out of sand, it will look like an acorn at some point because the sand adheres to it and looks like it has a little covering. This one is gregarious, meaning more than one will appear at a time, perhaps, and it's associated with broadleaf trees or cedars. Some puffballs are considered mycorrhizal, but not too much is known about which ones are which, at least not in my documentation. I was surprised to find out puffballs have an odor. In this case, this one does have an odor. So I've never tested a smelling of a puffball before, but now I think I have to. Some of these links are associated with spores, if anybody wants to go look at spores. This other genera, Bovista, I don't think I've ever found them myself, but we know they are in New Jersey. This case is fairly colorful, and you're not going to mistake this for a regular puffball because this case is sort of yellowish. And then it will turn colors on the outside. This one is more likely. This one's kind of strange because you'll see the outer peridion just sloughing off in big chunks. And this one will disconnect from the earth and blow around. That's why it's called the tumbling puffball. There are a bunch of other Bovistas. Some of them are named after their spore shape, the longospora. The minor one is supposedly rare, so we should look out for those. It's embedded in soil, has an odor that's sweet. The plumbia is going to have a bluish gray on the inside, peridion. And then there's disputes about which ones of these are separate or not, the bacilla or the longospora, whether they're one and the same or variation. So that's also one of those debatable things. This genera, I was very interested to find because I'd never seen it, and I remember Nicole found it, amazingly, just a few miles from where I live. Probably I've never seen it because it's usually on a trunk 4 to 15 feet above the ground. I don't know if she found it above the ground or it was from a fallen tree that was still moss covered or not. But this one looks very unique. You can see a little pinkish stuff on top. So when you see this little pointy thing, very small, this is her hand, very small, you might consider looking this and posting it because we only have her as a record. If we go into the bigger ones, the Bovista, the Bissett book talks about that as being baseball sized, though I don't think they're that big. The Calvatia is talked about as being basketball sized. There are 3 in my area, New Jersey. There's the one that has the purple spores on maturity, the cyathiformis. There's the grainiformis, which has a particular shape, looks very much brain-like. And you can tell by the gleba. Again, it's easier to tell some of these things are once they have matured. The gigantea is almost very round. It has a particular shape, in fact, that's very round. And that's the one that gets the very biggest. The other ones that are talked about in the Bissett book, I have not seen. One of them is talking about the Gulf Coast because the book does cover the East Coast all the way down. And there are some iNaturalist postings of having the giant western puffball in New Jersey. I look at the pictures and they do seem quite different than our puffballs, but it's not in the Bissett book. So if you find a western puffball, go ahead and post it onto iNaturalist or MO, Mushroom Observer. The Fragilist, again, is in the book, but it goes out to Michigan, and he's trying to cover Michigan too. The rubroflava we haven't found here, but it'll have another unpleasant odor and a color scheme that will be different than ours. Normal ones here on the Calvatia. This genera found by Maricel is one of the ones she had given a presentation to us years ago in person where she followed this puffball in her neighborhood where develops underground with the whole, basically the top part pointing down. And then when it matures, it flips up. And again, it's called an acorn because it still adheres with sand. So it looks like it's upside down when it's really right side up. But it's interesting the fact that it will develop underground and then invert when it gets out. The bigger group of genera is Lycoperdon, which I hope that also you'll share a bunch of examples tonight. I tried to explain some simple things about this newer one I haven't found myself, the Americanum. Most of these seem to be posted as being up in Canada, but that might be because they're out looking for it and separating it from the regular perlatum, which is a normal one that we'd normally think of. The long stem one you should be able to identify once you see something that's very long stem and is still like a Lycoperdon. Look at the spines on the perdims. Some of them will come into a pyramid shape. Some of them will fuse together on the tips like spines on these pulcherrimum. The pratense is yellow with spines. The subincarnatum is going to be reddish or purplish and on pink background. Again, something if we ever see it, we'll be able to identify it. And again, there's some dispute between Umbrium and Mole, whether they're one and the same or not, depending upon what author. But they'll have the spines turning dark. And also the nigrescens also appears to have dark spines. Some examples from Marisol of some of the ones, the marginatum, which is the peeling one. You'll see this whole thing slogged off and just left this one piece left. So it's big pieces that just fall off as opposed to just the spines. This Arachnion you can't really see too much. Back. But these have fairly long spines. The picture wasn't that clear unless you really blow it up high. This is one with a pedestal, which obviously looks much different because it's a different kind of shape, very tall as opposed to roundish. That one also has an odor, according to Marisol. This cork star one, we showed the picture of that black cat inside a earth star. Well, this one looks like an earth star too. This past the puffball will break open like an earth star. Instead of breaking open with just a little pore like a Lycoperdon or just in big pieces and open up like a Calvatia. Then there are stalked puffballs, the Tulostoma. Supposedly there's 250 species. They didn't say where, but I assume they meant worldwide. But a lot of them will require DNA to get down to what species they may be. You'll see postings on bromallii, which is what Marisol posted under, but that supposedly is the European name. The correct name probably is rufum. It's also a sandy soil. So again, down the pylons in the South Jersey. Calostoma, another unique one. This one goes into the order closest to the boletes. This is a mycorrhizal one. And we have the reddish one, gelatinous layer. We have the yellow one, again, gelatinous, but with a collar. And then we have the ravenellii. They are similar, but they will not have the gelatinous layer or collar on these. And these would be told apart by the which is that little top part on the thing where the spores would come out. There's not much online for keys, but there are at least two of them on the microportal, and I don't know how out of date they are. And that is pretty much the end of my presentation. Do we have any questions on my presentation?
I joined a little bit late, so
okay, I showed you all your pictures.
Yeah, well, did you show Leptocollus muscorum?
Say that again, Leptocollus?
Leptocollus muscorum. It used to be a Lycoperdon. It's actually on the cover of the Bissette's Puffball book that just came out within the last year.
All right, here's the Puffball. Which one is it? The top one?
Top photo. Yeah,
Okay,
that's
I didn't
it.
look at the credits. All right, so let me
Yeah,
look at
I
the
have.
Lycoperdon.
Now that name may have changed again, but
Well, that's what I'm saying. These are the ones. Which one of these is the one you're talking about? I didn't quite get the word you hear.
Muscorum is the species name.
Muscurum, yes.
Oh,
That
it's
one, let
there.
me
It's
have
on
some.
the list.
I
Yeah,
didn't put it on the
fifteen.
Muscurum is there. It's considered rare,
Oh,
5%.
you want to see it? It grows in my yard.
Yes, if
I'll
you have
put
yours,
I'll
let me
put
go bring it up.
I'll put a link in the chat
I probably have
for
the
an
link already.
observation.
I've looked them all up. I just didn't
Oh,
want to
okay.
include too many of them, but here's your rare one.
Yeah.
I didn't see anything that I can immediately say, oh, that's different, that's rare. So why don't you describe that for us? What's other than the fact that it's a very
It's
tall
fairly
guy?
smooth. I believe it has a pore that opens up on top, but it has a stalk. They always have a stalk.
Okay, very
Um,
tall, yeah. Does
yeah,
that mean it's a sterile base? I mean, you cut open that bottom part. What does it
I
look
think
like?
it's a sterile base. I haven't seen it in a couple of years. It hasn't come out. I've also found it in a park, um, in a grassy area of a park that's only a few miles from my house.
Well, here it looks like it comes in Morse.
Yeah, that's what's a mossy area of my backyard lawn. Sometimes it's in the moss, and yeah, usually it's in that area that's kind of mossy. Hmm. Yeah, yeah, it does grow in a mossy area of my lawn. Now
Do you remember?
the other spot where I found it, I think was just grassy, um, but that was a long time ago. That was like 15 years ago, so I don't quite remember.
Do you think
Yeah, so it's
these are?
actually. Oh, God.
Do you think? Do you think these are saprobes, all the lycoperdons that you are familiar with, or some of them
I
are
think they're all
mycorrhizal?
saprobes.
Okay.
I think these are all saprobes. But the fungus might live, amongst the grass roots or moss roots or something for a long time. For a while,
Did
I
you
was
take
finding it
this?
almost every year.
Did you take this over a period of time, or is
Yes.
this the same? Okay.
Yes, over a period of
15-day
time. It
period.
probably says in the notes,
Yes, 15 days.
15-day period. Yeah, it grows right in my backyard, so I can go out when it comes out, I can just go outside every day and see what it looks like.
I'll continue to take photographs of it because we need more photographs of it because we hardly have anything out there.
Yeah, I guess I, I, you said it was considered rare. I guess I don't
That's
find
what
it.
he put in the book
Yeah. Oh, okay, good.
because it may mean he just not, people aren't posting photographs. So underreported or rare, we can't tell the difference.
I don't know if I've ever eaten this. I probably should try it out just to say I did. I mean, all of these puffballs are edible when they're white inside.
Well, I think some of them
It's,
didn't say they did not know.
yeah, they
Like
actually say the edibility is unknown on Muscorum.
Yeah, well, I need to eat it so that we can say that it's no longer
Yeah.
unknown.
Sounds good. Yeah, just try a small bit though at first, of course.
Oh, I'm sure there's nothing wrong with this. Is the kind of thing
Yeah,
that if it was poisonous, we
you
would know.
believe that. Yeah.
Yeah, we would know.
You said all the puffballs are edible while they're, white or whatever. How many puffballs have you eaten? And are they any good?
Bovista peel is not bad. Um, the big Calvatias are good. Calvatia gigantea is excellent. You can slice it into half-inch or thick slices, bread it, coat it with egg and bread it and fry it and make it like eggplant parmesan. It's very good. Makes a nice casserole. Um, what else? Lycoperdon perlatum is it's the flesh is not very dense, so I mean you can eat it. It doesn't taste that bad. You can Apioperdon pariform. I've eaten that. It's not bad. They're
Haven't
small
eaten the
though, you know.
Calostoma, have you?
Oh, well, yeah, I don't really consider that a puffball. Yeah,
Okay.
and
Well, they consider
that's
it
kind of
puffball. Yeah.
a alien puffball, the puffball
Yes.
from another galaxy or something. You know, it's that's a
Well,
very
yeah, and
strange genus. I guess they
probably
call it stocked puffball in aspic or something like that's probably a Gary
Yes,
Linkoff
indeed.
name.
Aspic.
Yeah.
probably you could say all the ones stemming from the Agaricales order, but not
No, you
because
can't even say that because I don't know about this
Okay.
Arachnion.
I should qualify this that if you consider the Scleroderma genus to be those fruit bodies to be puffballs, then that would be the major counterexample because those are poisonous.
Well, we call those just Earthballs. We don't, they're separate from puffballs. So
Most people call
yeah,
them Earthballs. Yeah.
yes,
Hmm.
yeah, common name. Yeah, so yeah,
Hmm.
Earthballs we don't eat. The others, or I still make sure the species is known. Okay. Especially since
Yeah,
they've
I
all been
have
used
not
as
eaten
around.
Tulostoma. So,
Yeah.
oh,
Okay.
I
Yep.
found Tulostoma. It grows. A friend of mine has a barbecue in his backyard, and he made this like little patio, and he made the patio be able to drain when it rains. So he's got like sand underneath um, the stones that he put down, and then he's got like some space in between, so the water will drain off the patio and go into the sand. And Tulostoma grows between the cracks of the rocks in his little patio at his barbecue. I had them sequenced. I think they were rufum. I'm pretty sure it was rufum. Tulostoma rufum or rufa. I guess it's ru. Is it rufa or rufum?
It's
It's
Rufum. You know, knowing
rufum.
that.
Yeah. So um, I did not eat those.
Okay.
I guess you can consider those puffballs. Um, they don't. They're not very appetizing. They're just like this round thing on a stalk, and then it's just a spore mass that I don't think it. I don't think it ever really goes through a stage that's more or less like a gleba. I think it's just like powdery spores almost from the start.
the besets say inedible for Tulostoma rufum. So
Yeah, that sounds right.
yeah.
I mean, you had the book too, the Hunter?
Yes, yeah. I have
Okay.
some photos in the book as well.
Oh, yes, I kind of remember that. Yes, when it first came out. Yeah.
Yeah. I have a few things to say about the book. I love it, and I'm glad that you did this too because it gave me a reason to look at it more. One thing I wish it talked more about was seasonality, and then also range. I would have liked more info on that, and I feel like with INAT and stuff that we should be able to get some more range info.
Yeah, if we post more pictures, and
Yeah,
some of these things are so common, we don't think of posting them.
yeah. It did surprise me how many they said were quite common, and I was like, "Oh, really? Wow, I didn't know."
Some of them say widely distributed, but that doesn't mean it's easy to find. It may just be associated with a small type of habitat, and we just don't know what it is.
Yeah, quite a few of them have rather sporadic seasonality. I mean, you can find them in late spring all the way up until October.
I guess, yeah, yeah. And then the other
Now,
thing,
the Calvatias are late though. They're late summer species,
yeah. I mean, I think I'm
early
most
fall.
like right late summer, early fall, but maybe I'm yeah just most familiar with the Calvatia or Calvatia. There's one other thing that struck me. I can't think of it, but yeah, this is good to look at these.
I
Oh,
was just imagining that.
there's a nice key in the book as well. So Sue mentioned some other keys, but there's also one in the book.
Right.
Yeah.
Yeah, and this book is not necessarily cheap. It's a $50 book. Um, And that's quite a lot to spend on a book that's a very limited range of things that aren't edible, like earth stars and stinkhorns, grown-up stinkhorns.
But there's some beautiful pictures.
Yeah, thank you, Dave and Hunter.
Yeah, you gotta love them.
Yeah.
I just imagine that I've walked past 1 million of these acorn-looking ones.
Which ones?
Yeah.
The Lycoperdon? Or?
The ones that, like you were saying, that Maricel had, they flip over and all that, and they,
Oh, the inverted one? The one that
yeah,
Yeah,
starts
and sticks to them to make the acorn top.
this one. This one, yes, the one that isn't. You have to unbury it and see it's upside down, and then watch it go up.
last time we were in the woods, we were like walking through this section, and there was 1 million acorns on the ground, and now I'm looking at this thing like, yeah, any one of them, I suppose, could have been.
Yeah, I guess you'll touch it. Once you touch it, you'll know.
Yeah.
Yeah, you have to be in the sandy area. It seems to be a lot more interesting things in the sands than I would expect it.
Are you also covering Earth stars?
No, I was just going to cover the puffballs.
Okay,
Um,
yeah. I just wondered because I had
Yeah, I even stopped
some
at the
interesting
Earthballs. I
ones
think
of
that's a
those
little
I
too
found.
much because we do want to get to member finds also in the same evening. I know Igor gave us a link in this chat, and I don't know if that's a documentation. Is Igor on? He wants to describe what this is. This is
Yeah, basically, it's a huge study of Lycoperdaceae that came out from China, but basically, it's a global. It has global coverage, so it gives you all the genera that are currently in Lycoperdaceae sensillata. It's both phylogeny, taxonomy of the genera and species. I don't think they have a lot of coverage from North America, but I think it gives a background. I mean, if you really want to study puffballs
shows you the
and
vasicellum,
allies,
which sort of disappeared.
pardon
Yeah,
me?
and it's Bovistellum. Yeah, the
Yeah,
vasicellum seemed to disappear. And this Bovistella I did not mention, but it is also one of the ones that's is still there or had been moved into Lycoperdon again.
yeah.
So okay, so,
So
all right, so we got that
it's a
mark.
long paper and it has a lot of phylogenetic trees, but it also gives sort of a key to morphological traits and spore shapes and appearances. You'll see those pictorial presentations. It's a nice paper.
Oh, very nice, very nice.
Yes,
Here's the tree. Yes,
and this is just the
Calvatia's down
beginning.
here.
Cool.
Yeah,
Yeah, some of them are global. Some of these are spiky spores.
right.
Yeah, the few spores I've looked at from Lycoperdon pulcherrimum, they were really decorated, really ornamented, really fun to see.
Yeah, like Calostoma spores.
Are they really cool?
Yeah, they're super spiky. They
Mm.
kind of under the right magnification because they're the right size, and they, like, if you get too many on the slide, they all look like little gears connecting.
Oh, cool. I can't imagine even getting a Calostoma home in one piece at all, but I guess it can be done.
They're pretty hardy.
That's surprising. Yeah.
So
Interesting.
is the hymenium the head on those just the surface of the cap?
The Calostoma?
Yeah.
No, they're you
Oh,
kind of poke
the
them in
spores
the
are inside. Okay, I'm
yeah.
sorry. Yeah, yeah. So you can squeeze them out. If it's mature, they'll just come out like a puffball.
Oh yeah, you just every time I see them, I walk by and I can't help myself. I gotta just like push it like a little button.
Oh, okay. Because I usually leave them alone. They seem, kind of messy. Well, I usually
Same.
find cinnamon
Messy.
and
The
I usually
collared
find
ones,
cinnamon.
the collared ones are like, they look wet, but they're kind of dryish.
Yeah. I think what I should do is put a couple of microscope slides in my plastic containers, and if I find one, I can just collect spores right in the field.
Oh,
Because it sounds like
easily.
they have interesting spores. So I'm only familiar with spores from a few different kinds of puffballs, and I know that sometimes it's easy. Sometimes Lycoperdon perlatum and Apioperdon pyrifum can be confused, because sometimes Lycoperdon perlatum grows on well decayed wood, even though it supposedly doesn't. I'm pretty sure it does. I've just had a discussion with Walt Sturgeon about this. He agrees. Um, so look at the spores, and that'll tell you the difference, right? Well, the spores on Lycoperdon perlatum and Apioperdon pyrifum, I think they're both like 4.5 microns wide. They're globose. But the one species has really, really, really small projections, warts, and the other one doesn't. And it's really hard to see the ornamentation. I forget which one it is that's ornamented. So,
One of the discussions in the chat is Toby brings up that he shares a medicine book that Calvatia gigantea is good to eat, and the spores are flammable, and we use as fireworks. And the dust stops bleeding wounds to stop the flow of blood. And Hunter says the book says burn the spores to calm honeybees. Okay,
Yeah,
any
that
other comments?
anticoagulate quality, yeah, I think is definitely a real thing.
Any other comments before we start showing perhaps Hunter's puffballs? Okay, let me stop this.
I've shown the first picture to before. If we can open it.
It's not. Yeah, it's trying. It's not getting anywhere.
Yeah, that's weird.
Oh, there it is. Okay.
Oh, there it is. Okay. Yes, I've shown this before. This is in the Bisset book. I took it. It's Lycoperdon pulcherrimum, and it has spines that are fairly long. So that's the exoperidium, and they're 4 to 6 millimeters long, and then the tips are often fused. And then you can see that they eventually are shed, and basically disappear. But
Do you know how long it takes to go from full to that? Because one of the things with identification is saying, well, how can I wait to find out, is this going to slough off in little pieces or uh, not?
yeah,
On a lot of these lycoperdons, I don't know how long it's going to take unless you find a big group in different ages.
yeah, I think it's really going to depend on conditions. This was, I'm pretty sure, a drought year. I mean, it was two years ago, I think, and I think we were really pretty deep into drought. So maybe the dry conditions contributed to it. I think it's possible that you don't get to see that always. That maybe they're like eaten by animals, or maybe if there's a lot of moisture, maybe they don't they decay in a different way. I'm not sure, but yeah, I don't know.
Great photo.
Yeah, it was really cool to find these. It was really beautiful, and pulcherrimum means beautiful. So this is the long-spined puffball or spiny puffball.
Was this in a deciduous area? What is this material
Yeah,
on the ground
it's
here?
spruce. It's a kind of a large stand, very large stand of once planted Christmas trees that are way overgrown, many years overgrown. So there's white pine, but there's also a lot of spruce, and these were in, I think, spruce. And I'm terrible with conifer identification, but those look like spruce needles. Yes.
It could be. Yeah.
Yes, I would
Right, they
agree.
look like flat.
So they're just saprotrophic with on the needles there, or what is that?
They are, I had not heard Sue mention that there are a few puffballs that are mycorrhizal, but I assume they're saprophytic on something here. There's horses in the area, so maybe hard to say what they're eating. I don't know. But I mean, they're growing from the ground.
Yes.
Do they come up
Yeah.
regularly, or is this just a one-time thing that you saw them?
No, I did see them again in the same area this year. So yeah, but they weren't in the various stages like you see here, which is kind of nice.
Perfect. That's great. I didn't find a good one on online. That's great.
It was right there in the book for you.
Yeah,
Um,
well, I don't want to look the book. I wanted to use ones that were found in Jersey. Sorry.
and so
Okay, and this one is
yeah, well, I am not sure. I'm going to call it, I want to call it today Mycenastrum corium. Um, but it's a real anomaly. We found this like
that
it
the
was
mosaic one. Is that the mosaic one?
yeah, it's the one you talked about it. It's the one I think that it sheds like an earth star, you said, which I hadn't read at all.
Oh, okay. The Mycenastrum, something like that. The strand.
Yeah, Mycenastrum, right. But it may not be that. My other guess would be a Western Calvatia. But the weird thing about this was during morel season. It was like April 30th, and it was in a traditional hunting area that it was real sandy soil, wide open. It's hard to describe this area, but it's it was just sort of really weird to find this puffball that generally I find them in the summer and fall. There was like maybe the first morel I think we found like our first morel in the this part of Illinois, right around the same nearby, relatively nearby. But I'm not sure for sure what it is. I do have a cut picture of it too. Yeah, I mean, and I didn't really have, yeah. So I don't know. I thought the cut picture, it kind of reminded me, this is it, of the cut picture in the book, although mine is fresher. It does seem to have that pretty thick outer, that thick endoperidium, I guess.
Or
Yeah.
okay.
So, but
The book of is the Mycenastrum. It tends to be associated with cow or horse manure, but you weren't near cows and horses, were you?
no cows, but yeah, there's horses go through there.
Okay.
But yeah, there's no cattle there. So that's okay. That's good to know though. So yeah, I don't know. there is an equestrian area right there, so maybe. Maybe. Dave, do you have
Just saying,
any
it's got
thoughts?
this mosaic pattern. Some of those books.
I know the Western Calvatia always has a real wrinkled look too, but it being April 30th, I'm like, nah, it's not. I don't think it's that. Um,
Do you have the Western Puffball out by you?
Not generally, generally no,
Okay.
but I think an anomaly that yes, I mean I think it has been found, but it's generally not. So yeah, I don't know if anyone has any thoughts, but it might be one or the other.
Well, upload it and see what iNaturalist thinks. See what
Yeah,
iNaturalist guesses at.
that's a good idea. Yeah, I will do that. Thank you. Yeah, and
Because
then
it looks like that mosaic one. But yeah, it's like if we only knew what it looked like when it was matured, what color the gleba is going to be, uh, that would really
right.
help.
Yes.
And it's like, oh,
Yeah, so this is another contribution I made to the side book. It's Calvatia rubroflava, and I mean, I thought this was like the rarest thing, but in the book they say that it's scattered. Well, widely distributed. Okay, so and this was in a landscaped area, in a very fancy, at the entrance of a very fancy estate, and so I think it must have come in with some landscaping material. And it sounds like it likes to grow in gardens and grassy areas and woodlands, but um this is it, and it's got rubroflava for this red orange staining that you can really see on that first one. It is edible as long as it's white all the way through when fresh when you cut it open. And its common name is orange staining puffball, and it's pretty cool. And it is a Calvatia, at least for the moment.
does one have an odor? I don't remember that.
Yeah,
Yes, odor, strong and unpleasant.
yeah, and I kind of wanted to add that I think all puffballs, when old, have a pretty bad odor. I think that's just kind of when they're old, though. I mean, Calvatia, when they get old, it's like, oh, really not a good smell.
Is that after they've broken open or they're still
Oh,
closed?
not necessarily. No, just when they're, inside, they're starting to change. Yeah. Yeah, it's you can and it's kind of happens pretty quick. The smell turns from, like not unpleasant to like offensive. And it might still look okay outside. And then I got
This one?
oh, what's that? Oh, yeah,
It's tiny.
I love this one, and I may have shown it before. Clearly, it's really overexposed, but I believe it's growing right out of that pine cone, which, none of these in the book say anything about doing that. I think this is that, and I think I may have shown it before, that is it curtisii?
Yeah.
Yeah, that's
Long spines, yes.
my guess. But and so yeah, and so I guess these have spines, but they aren't as long as the pulcherrimum, and they aren't fused in the same way supposedly. But I don't know for sure. This is a very old photo.
It's
Does
the end of
anyone
your
know
puffballs.
that plant, by the way?
Yes.
What is it, Sue?
I don't know what it is. It looks like a snake plant.
Oh, the
Yeah, family.
plant right there. I think that's rattlesnake plankton.
Oh, okay, cool. Yeah, because that's a good.
Is this rattlesnake plankton?
Might be, yeah, I think
Yeah.
so.
Yeah.
I think that's a pretty kind of rare around here, that plant. So cool, thanks.
Yeah, we have a lot of it around here.
Do you? It's
Yeah.
a pretty one. I like it.
Yeah, it is nice. It's in the orchid family. That's in the orchid family, isn't it? Yeah.
Mm, that's interesting.
Pretty common in North Jersey as well.
Yeah.
Ah,
What kind of cone is this? Is this a spruce cone?
white pine, probably.
No, not
I
white
don't think
pine.
that's white pine.
Hmm.
The scales are larger and more separated.
Hmm. Hmm. I don't know. This is a really old picture where I thought it would be have been taken. Where I thought it had been taken, it would have really had to have been white pine. But oh, you can see some looks like you see the needles on the left of the pine cone. There's some pine needles.
Those
You
look
see?
like spruce needles.
Yeah.
Yeah,
Okay.
Dave, this is a spruce cone, definitely. Look at the shape of the individual, how do you call them? Scales.
Yeah.
Okay. This must have been somewhere up in Wisconsin or something, not where I had remembered it being, most likely, because we don't really have, except for that Christmas tree area, I don't know where else I would have found this in spruce. So now I'm thinking maybe up there. I don't know. So yeah, well, thanks for letting me share that. That's sort of it. I think I might have, like, I could throw one more in, but I think that's all I sent. Is that right?
Yeah.
Okay.
Yes.
Cool.
Yes.
That's
And just to clarify my mycorrhizal statement, Bissett book talks about the colostomas being mycorrhizal, at least those. And I know there's others that are associated with hardwoods and conifers that they of the species, but I don't know whether that's because of the saprolite nature or not. But definitely the colostomas.
very cool. I never see those around here. I've found them, but not in my state ever.
Does anybody have any other puffballs they'd like to show before we go to member finds?
I can share
Hunter,
one
do
that
you
I
want?
think that
Okay,
I've
good.
misidentified.
That's your puffball,
Let me
The dyers, so pretty.
let's see. Where is it? All right, let me share here. All right. I didn't look to see when I observed this or posted it, but clearly it's growing in lawn. It might have been even in my backyard. So based on that, what color would everyone say that the gleba is?
It's
My
tough.
screen looks gray.
Yeah, it
That's
looks
like
gray, but maybe
gray.
I don't see purple.
Could be
tinge,
purple.
maybe. I think that's the one they consider it purple inside.
You think that one's purple?
Yeah, that looks like probably cyathiformis. I think. I don't know. I might my concept might be faulty.
All right.
Those two species are really hard to tell apart.
They are, and
Cyathiformis
the
and
outside
Cranioformis.
looks like kind of brainy, right? This cranium brainy.
The
They
craniformis,
both look that way.
They can both look that way. All
craniformis,
right.
Yeah.
craniformis is brown on the inside when it matures, though.
Yes.
It goes
That's
yellow brown to brown, so that's definitely not
yeah.
brown.
All right.
No,
So I guess
that's
we
what
had to
they
choose
call
between
purple, I'd
brown and
say.
purple.
All right. It's kind of subtle to me.
Yeah.
A
So
lot of colors applied to mushroom descriptions are sort of subtle versions of the given colors.
yeah.
So I think this is a good example of that, really.
All right. Well, then sounds
Do we
like
have
I
a
did get it
size?
right.
Do
Cyathiformis.
do we have a size? Do we have a size of the
No, I just said it was growing in lawn. I didn't give a size, sadly. When was this? Oh, yeah,
just
this is
because the
back in 20. Yeah.
Fabulous
It
has a
was
dark
pretty good,
purple one too, but it's
you
smaller.
know,
Oh, really? Oh, cool.
bigger than a grapefruit, I would say. Or maybe at least the size of a grapefruit. You know, but yeah, so all right, so that was one that I
Love
thought
the
was
purple.
interesting.
I love when
Um,
they take on
We
that
did
mushroom
have
shape. Like it looks like a mushroom. I mean, like a stem and cap shape.
yeah,
It's funny.
yeah. And then I don't know, is this marginatum with this peeling going on?
which one?
Oh, wow. That's nice. Mm.
It looks like it's sloughing off in plates.
Yeah. This this comes up in the same spot every year. I see it every year, but I think they're so beautiful. I think they're really cool. There's one of the
Where they tell you to go look at this and see where they're pyramid shaped. You can see that these are look like individuals.
yeah. And hickory growing nearby.
Now, were you thinking that this was the one like Hunter showed that'll?
No, marginatum. Mine was pulcherrimum.
Yeah, I don't think it's the pulcherum. I don't know. I thought maybe it was this one, but
Yeah, the pulcherrim has the well, I know the species from seeing Hunter's observations, but the distinguishing feature really seems to be the very long hair-like spines that sometimes fuse together at the tips. There's another one that does that too. Isn't it echinatum? Or
Right, and that's not in the book, in the Beset book.
yeah, but that one, but the spines are not so long and hairy in
Yeah,
echinatum.
that could be the one on the pine cone too, I think. I can
Oh, okay.
I don't know though. Mm.
Well, what Lila has looked like what's in the Beset book from marginatum. The
right.
same
Does it?
pieces of flowering.
Okay. Yeah, here's another one that might be the same. Yeah, I think it's the same thing. Different observation, just an older one, not as photogenic. There's a younger one before it starts. All right, so you think that's marginatum. All right. That's what I thought too. Oh, yeah, I don't think I have anything else that's interesting. No, that's it. Okay.
Okay. Any more puffballs to show for tonight? Anyone?
I think it's pretty hard to
Otherwise, we'll go.
ID these. I mean, that last one Lila was showing, I feel like it could be a couple things in the book, but
Yeah, I'm always scratching my head with them.
yeah. All right.
Have we looked at an observation of Bovista yet?
Yeah,
I had some in my presentation, but you
yeah.
want to share them? We'll look at some more with Vista. I'm not familiar with Vista at all, other than what I put in the presentation.
Okay.
Do
So
you want me to?
we haven't looked at any observations, personal observations of Bovista then? I'll
Nope,
send them.
let's look.
The ones that, yeah, I had in the presentation.
Okay.
Got Brandon's turning on share. It's slow. I noticed that all day today on iNaturalist, everything was loading very slow. I think they're getting like Mushroom Observer.
So do they only call Bovista true puffballs?
I don't know. I think that's probably just they decided to call something that on iNaturalist to differentiate it from Lycoperdon and stuff. Probably. I don't know.
I'm curious.
But I don't really have much to say about this. I only have it to the genus Bovista, mostly because I'm
I
pretty
just want to
unfamiliar
see
with
it.
them.
Is your PowerPoint still up or whatever?
No,
No?
everything's closed other than empty tabs.
Okay,
Okay,
cool.
open. Well, all right. I'm trying to enlarge it, but it's just hangs.
Is this from the Pine Barren?
Not the Pine Barrens proper, but a very similar ecology.
Yeah, super sandy, huh?
Hmm. Yeah, this is kind of well, around where I live. It's the edge of the inner and outer coastal plains. I've shown this here. I showed this here last year when I found it. Um, but yeah, pretty similar ecology to the Pine Barrens. Um, Not much to say about it. You know, it's small. If you can catch the little tail that it has,
Oh, it's so small.
yeah, there you go. And it's got a little white rhizomorph that's pretty tough. And I didn't get all of it. You can see it kind of breaks apart as it gets deeper into the sand, so it gets kind of wispy. But right up, kind of close to the bottom of the thing, it's pretty sturdy. You can dangle it at least by the rhizomorph.
Is that how you identified it to genus Bovista because of the rhizomorphs that tie it down to the ground for a while?
No, but what I always find fascinating about things like this is the spores look very similar. The spores is how I identified the thing because they have
Oh.
that pedicle. That
Oh, the pedicle. Oh, okay, right.
the spores kind of look like that last photo we looked at.
I think the strands on the base of the fruit body that sort of anchor it to the ground for a while. I think that's also a Bovista.
I do as well, but there are Lycoperdon that have rhizomorphs. Um, in fact, that's how.
Apioperdon does. Apioperdon
Mhm,
pyriform does, but that's on wood though.
sure.
Yeah.
Yeah, and I guess we're not able to zoom in here, huh?
Yeah, I can't get anything to zoom today. And you're supposed to click on it, right? I haven't forgotten that, right? Just click. It just doesn't want to do anything.
Maybe give it a second, you know.
Okay, be patient. Yes. Click on and be patient.
But if it doesn't end up coming up, these spores are globose, so they're nearly, perfectly round. Not perfectly, but they're globose and not subglobose. And they have, oh, there we go.
Where
You can just click to zoom.
left? Right.
Oh.
Just to here. Oh, okay, there we go. But you wanted the other one probably, right?
Probably, but you can see it. Yeah, so the one by the 4, it's a bit out of focus, but you can see that kind of line coming off of the almost globose spore. I think that's called a pericle. It's not a peridial pedicle.
Pedicle, I think.
Yeah, and you can't see it as well in this photo. Maybe the first photo that we were able to zoom in a little bit on, you can see it tucking in. That pedicle reaches almost 10 microns. Yeah, there you go. So this is at 400.
Whoa.
Yeah, this is at 400. At the largest, that little line coming off the side, or where it is attaching to the gleba, reaches up to 10 microns. So it's in some cases larger than the actual spore itself.
Huh.
That is excellent and weird.
Yeah, and I think that is a very distinctly Bovista thing, along with, like Dave said, the pretty strong rhizomorphic network. But I think the Bovista, they all have this, if I'm not mistaken. Again, I don't know the genus well enough, but I know that was when you're looking at puffballs. Does it have this little tail coming off of it, or not?
Well, that's good to know. And then the exoperidium on the the like non micro, is that what it looks like when it's fresh? Or is that more mature, or are they brown, or this species?
Oh, sorry, I was muted. This is probably a mature specimen, right? This thing was probably white, uh, you know, before it bust open and started, releasing its spores and everything. Probably is too much. It could have been white at some point.
Did you get a species on this, or no?
No.
Okay, just curious because I'm really not familiar with these at all.
Yeah, me either. And I think I think the name Bovista predates Lycoperdon, if I'm not mistaken.
Yeah.
So I think maybe a lot of these things spend time in Bovista. A lot of these puffball type things spend time in Bovista.
Does that word have something to do with cows or something? Bovine or
Yeah, I think it does.
Bovine does, yeah.
And I think a lot of times the bovistas are found in like cow pastures.
hmm.
But yeah, just wasn't sure if we looked at any of our observations of Bovista. That's pretty much all I know about them. I think this might be the only one I've ever confidently ID'd to the genus, but yeah, I looked at several species, didn't really come up with anything good.
So maybe because it's like one of the older names, that's why they're called true puffballs, maybe. Like is the first name.
I would track.
Maybe.
That's all I got for the puffballs.
Yeah, well, I wouldn't buy the Bissett book to figure this one out because it doesn't seem to match anything. The Bovista pilot does say it's globose spores, and some of the other Bovistas are not globose. They're subglobose to oval. So
Do they all mention that pedicule? Pedicle?
I don't even see it mentioned. They on the pila. It talks about uh, short broken pedestal cells and capillary threads, mostly 11 string wide, but up to 13. Dark brown, frequently branched, tapering to a point, thorn-like spines. But we don't even have spines on this, really, right? This is.
Are you looking at the spores? Or are you looking at the
I'm looking at the
rhizomes?
microscopic features, and I don't know why they're talking about spines on the microscopic features.
Oh, they're ornamented spores.
Yeah, but they don't say that words that we normally see.
Yeah, and
Um,
Brandon's didn't look like it had
yes, they
spines.
said the spores are thick-walled, smooth, pale brown, and short broken pedestals, but they're talking about these branches and thorn-like spines in the microscopic features.
Right. So are they talking about the spores, or are they talking about a different thing under the microscope, which is probably more likely, potentially
Yeah, it could be
the
talking
Gleber,
about the
the
threads. They
Gleberet,
maybe
or
has thorns on the threads.
Or maybe the pedicles they're calling spines.
okay, let's move on.
Yeah.
Pedicels are the threads on the spores, those little string-like things.
And
Oh,
pilot doesn't work because you're in sandy
right.
soil.
No, I know. I'm the spores. Right. But not the macro rhizomes or whatever hanging, dangling. Yeah. Okay.
Yeah, I encourage you guys to look a description up on your own.
Good idea.
Yeah, Micro Quebec did have a lot more puffballs than were found in the Bissett book, even for the East Coast. But we don't have any occurrences for them here. Um, so that's it from you, Brandon?
Yep. Once again, that's it.
That's it. All right. Thank you, Brandon, very much. First per Vista shared. Anybody else before we switch to member finds?
Toby put something into the chat. Can take a look at.
Still loading. I see it. They look like little pyramids that I can see.
Perlatum, probably.
We don't see the shape of the whole thing. But we assume this is on ground and not on wood. Yeah, I would assume it's perlatum. Anybody else want to offer an opinion?
Yeah, I'd call this perlatum. On the ground, it's ornamented, it's shaped like perlatum. I think
Shape.
that's what it is.
Yeah, this is nice. We do actually get to see the good shape of the spines.
Thanks.
Thank you,
Those were in
Toby.
my backyard during, I think, the spring.
Oh, really? In the spring?
I think so. I have to look up the date on the picture, but yeah, it definitely wouldn't be the summer to have all that those dead leaves. So I'm pretty sure it must
Well,
have been like early spring, but I'll check. But it was definitely in my backyard.
that's good.
Are those good for eating?
You can eat them.
When
You
they're
can?
white inside.
Yeah,
Okay.
they're not the best of the puffballs, but they're okay.
Well, that goes with my cooking.
Yeah,
Not the
they're not
best.
bad if you slice them and fry them up and just and add them to scrambled eggs or something. They're not bad.
Okay, I can disguise them. I'll try that.
What happens to the spines when you cook them, Dave?
Um, Gee, I don't know. It's been a long time since I've eaten any of these. I guess they just kind of fall off, but there's not that many of them. It's not as sort of overbearing as like hydnum spines.
One thing like with Calvatia, I peel the exoperidium off. And so I think that's sort of the way to go with puffballs is to peel off that outer skin. But with these really small ones, I mean like the pear-shaped puffball, I mean they're so tiny. It'd be hard to do. But I think those probably cook down in a way. But if you can peel it off, I would recommend peeling it off.
What's the advantage of peeling it?
I don't know what the advantage is. I just know that's what I've learned. You know, I've been taught that. I don't.
Yeah.
Yeah. I mean, one, it's clean. You don't. You know, it's clean.
Okay.
Yeah. Uh,
Yeah.
but I think it's probably a texture thing with like Calvatia, but I don't know. I mean, it's a good question, but I always peel it off.
You ever heard
Yeah,
that
because
bug
I haven't
problems?
peeled them, and
Hmm.
I haven't noticed any issues.
Negative. Yeah. Okay.
Do we have any bug problems with puffballs? I can't say any picked any up that are covered with bugs.
No. Same. Mm-mm.
I wonder why they don't like it.
Yeah, that's a good question.
I don't think their spines are enough to keep them away. Okay, because they're very soft. Okay.
Probably, yeah, I don't know.
Okay. Well, thank you, Toby. And about eating. I guess we're ready to go to the emails. And just to give people who have been waiting a chance, Doug, are you on?
Can you hear me, Sue?
Yes, I can now. Would you like to go now?
Sure. Let me get my mind off of puffballs. It's something kind of different. It was
Yes.
the end of last week's. Chris asked about um, fossilized fungi, as I recall, and some people were curious as to what the record was, and I thought I threw in a couple of references here to recent papers. This one came out this spring. This one is from the Jurassic, which is about 160 million years ago in China. And it's got some beautiful histology. These are ascomycetes that are in the group, polyphyletic group of blue stain fungi. And these fungi do not use enzymatic extracellular secretions to degrade hemicellulose, cellulose, and lignin, like what we call over here white rot, brown rot. But they penetrate the tracheids and the rays of woody plants. This particular genus, Xenoxylon, is an extinct conifer, and they've got some just fabulous histology here. They know it's an Ascomycetes because of these pegs that they produce that have these sharp points that penetrate through the cell walls. And they also do not have clamp connections, which the next paper, which is in the same genus but a different species, this is from northeast Utah, maybe about 180 million years ago. It's got some references for those who are interested in the history of fossilized fungi going back to the Rhynie Chert and Cambrian times. And so it's excellent for references if you're interested in the earliest fossilized fungi. Most of these are found in siliceous deposits of permineralized wood. The second paper has some nice images of the clamp connections. That's how they know they're Basidiomycetes. And apparently the patterns of how they penetrate the and degrade the cell walls can tell you whether it's a white rot or a brown rot, even in these fossilized specimens, which I found quite interesting. So I just threw it in here because I believe it was Chris, if he's still here, he asked about.
Yeah, I was wondering because, you know, the way that they are, would, you know, most fungus I wouldn't think would fossilize all that well.
Yeah,
and these seem to be, you know, found because they were embedded in, you know, in some wood or something, which is interesting.
Yes, that's absolutely correct, Chris. The you know, a straight like mushroom cap is much rarer for obvious reasons of the fossilization process and the permineal, you know, the replacement of the living tissue with minerals occurs over millions of years, and you have to have very specific conditions for a let's say a mushroom like we see, like a russula, you know, to actually form a fossil. It's quite rare, but because these are embedded in this wood, they're preserved beautifully. And I think in the second paper, they also see a lot of bark beetle holes, and as you probably know, the beetle is the source of allowing some of these fungi to get in, and you know they wreak havoc in our western forests currently, but have done so obviously 180 million years ago, which is kind of interesting.
Fascinating.
Yeah, those who can't see, the red arrows are pointing to these clamp connections that he's talking about.
Thank you, Sue. It's really beautiful histology. They do thin section of this wood. You have to have specialized microtomes to make thin sections of fossils of any kind that are mineralized or silicified, you know, and they have to be, you know, you can't use like a microtome in a pathology lab with embedded in paraffin. These are very specialized machines to give you these sections, and the staining is beautiful. So anyway, I thought I'd just throw that in, and for interest. It has nothing to do with puffballs, but
yes.
Forgive me.
Do you have part of the evolution?
Do you have a link to the article?
Yes, we have them. I'll put them in the chat, I guess.
Yeah, he has them there. I just sent them.
That'd be great.
Thank you. a lot of things about this meeting are fascinating and wonderful, but I thought your presentation was spectacular.
Thank you.
Yes, Sue, that was really
Yes,
well.
we'll have a lot more coming up. Of course, it's the winter. That's when we start getting away from just random finds. Come on.
And if anyone's wondering about blue stain fungi, which I was, that means that it's staining the wood blue. So chlorocybora is in there.
Got it.
I'll put that.
Yeah, I was wondering,
Sure. Okay.
but I knew it wasn't like, yeah, cool.
All right, thank you, Doug.
Yes, you're welcome. Thank you.
Very interesting.
The only other person who sent in things to show was Hunter. Does anybody else want to go before we return back to Hunter with her other fungi finds? Okay.
I have one, but I'll put it in the
Okay.
chat. Let's look at Hunter stuff first.
Okay.
Oh, I think these are for next week, actually.
Well,
Yeah, we should probably save
next
these.
week. Do you want to save them for next week?
Yeah, I'm
We
sorry.
still got time though.
Yeah, I thought I stopped this send because I had it ready to go, and then I saw it was Puffballs, but I guess it went. So
Puffballs and member pines.
yeah, let's wait if that's okay. I'll resend it to you,
Okay.
so you don't have to hold on to it or
Okay.
anything. Okay. Yeah, because I don't know
Look
if
at Brandon's.
house we're gonna have a freezing cold week, so.
Yeah, so I'm calling this Cystoderma, and I'll kind of just talk while it's loading. Found this last couple days. We had a nice warm spell and some rain, and I finally was able to get out in the woods and found some cool stuff. I haven't been able to work on it, unfortunately, but morphologically, at least, it matches one of these cystodermella type deals. I called it Cystoderma amyothinum at first, but I don't think the cap quite fits for that species description. It's a very small thing. It's growing on pitch pine needles.
And in general, I've been trying to sort of get to know this genus. Can you give us, while it's still coming up, some like features of the genus in general or things?
Yeah, so I think it's however you want to describe the hairs on the stipe. Attenuate, I guess, or it's not quite strigose. It's these kind of tufts of hairs from below the annulus all the way to the foot, and that annulus, I think, for Cystoderma is supposed to be.
Oh wow! Look at that.
Yeah. Now, the one thing that I was reading about Cystoderma is that annulus is really supposed to be like kind of reflexed from this stipe there, and you can see this one. It's more of like it's a pretty typical looking annulus. It's not as kind of drawn away in this membranous fashion from the stipe with some of these Cystoderma, particularly amyothinum, which I was comparing this to. But yeah, you can see that ornamentation below the annulus is tufts of hairs. I don't know how you want to describe it.
Cool.
There you can see
Sorry,
it's growing on
what species name did you apply here? I'm very tired tonight.
No, I applied amyothinum at first, and then
Yeah.
and then I withdrew it because I don't think it's amyothinum. And if we can have a look at the cap, pine leaf
Oh, little.
litter. Yeah, very little.
Let me know which one you want me to blow up because it doesn't seem to be paging, getting the arrow.
Yeah, this is the in situ photo, and it does show the cap texture, which is going to ultimately be granular. With amyothinum as which was the first name that I applied here. The cap is supposed to be kind of radially wrinkled, but this thing is small, like you said, Hunter. It's probably immature. You know,
There's two varieties of amyothinum. One is deeply radially wrinkled, and the other is fairly smooth, I think.
Oh.
Yeah, there's two varieties, or maybe one that's not a variety, and then one that's a variety.
Interesting. Cool. Yeah, and I haven't done a ton of work on this, but I thought it was a cool find, especially for, January 7th.
Yeah, all of the cystodermas have amylate spores. If you find something that looks like cystoderma, but it has spores that are not amyloid, then you have cystodermella.
Right. Yep. And then look at how scaly the cap is.
Yeah. Yeah, I'm going to have a look at the species because I know these a little bit.
And then if you can zoom in on the cap margin on that last photo that you just had up, that's another cystodermella thing, right?
With
the gills, the one with the gills showing.
Yep. Yeah, the cap margin. So you're going to want to look at the underside.
Oh yeah, they have like stuff hanging off the margin, right?
Yeah, and this one I think
like appendiculate.
Yeah, there you go. You can kind of make it out there. But I'm kind of the opinion this is a little bit too immature to show some of the other features that you might be looking for. But I was just happy to, yeah, I think it's a cystoderma.
That's pretty cool. You just found that right now?
Yeah, like less than a week ago.
Oh, wow. That's great.
Yeah, I went out for like my first time in a while, go spend a couple hours in the woods and, walked away with 10, 15 species of fleshy fungi, which was interesting.
Nice going.
Well, thank you for going out there, Brandon. I hope I'll see you again next week with more stuff. Because we
Thanks
did
for
have
letting me
all that
share.
nice rain. Thank you. Anything else anybody wants to show for tonight? We still have a little bit more time.
I got an old Calostoma I could show.
Sure, why not? You going to share from your screen?
Yeah.
Okay.
Ooh, pretty one.
Yeah, these are the collared ones. And you could see how much is sticking out of the ground now. And you could see how big and this is probably broken off some.
Well, that's cool to see that it made it home.
Yeah, it's like a big piece of styrofoam. It's crazy.
Yeah, they're pretty sturdy things. Nice.
Ooh,
Oh, wow.
beautiful.
And
Yeah.
that's why I like Calostoma spores.
Yeah. That's worth taking it home for sure.
What's
That's
the
excellent.
magnification here? Do you know?
Oh, that's probably 400.
Yeah, that makes sense. So
But
even at
they're pretty large.
yeah, even at 400 though, you can make out those spines. I mean,
You
pretty
really
well.
can.
Yeah, cool.
Yeah. I just and just the when you peel one out, like you could keep digging for that thing. I mean, there's probably another 2 inches on that thing. And then yeah, like the spores on there, they're that like creamy, they're almost like the same color as like this the yellowish part on the like the collar. And you just take them and just spray them on the slide, and like that little dot that's on the slide, like that was just putting it right up to it and just giving a little squeeze.
Now, I was in and out early in the meeting, and I heard, I think Hunter, you were mentioning you can never find these things intact, or they always kind of fall apart. Is that what I was hearing? Because I think I have the opposite.
Messy is the word Dave used, and I agreed because yeah, they seem kind of gelatinous and gooky sometimes, but I haven't tried to pick them up or touch them, so maybe that's just an appearance, be misleading.
Well, one thing I I love this anecdote. He's not here this evening, but Jason, who's often here and hosts meetings, he shared an anecdote about this particular species with me one time when we found it in the Pine Barrens, and we find it in January in the Pine Barrens. We find it in February in very freezing conditions,
Yeah.
and they're always in really good condition, like they're never falling apart or anything. And he has shared this a couple times that he'll collect mushrooms from wherever, and there's a specific spot in his backyard where he dumps the mushrooms. Um, so it's a constant state of decaying mushrooms in this one little spot. Calostoma lutescens survives. It does not rot.
Oh.
And I think in some of the descriptions, doesn't it have a chemical that is super similar to antifreeze? I think we looked that up one time, and it ended up being true.
That is something kind of familiar. Yeah.
That
Well,
would
I
make sense.
have
I always
found,
find them in the winter.
I've only
yeah,
found them in the
I found
winter.
them in the spring with old ones from the year before.
Oh, wow.
They were kind of eroded, but they were still intact.
And not like rotten, right? Just maybe falling apart on themselves.
Not rotten, fairly intact, but some of the ornamentation, on the outer stalk was maybe a little bit weak, but no, they, from what I remember, they were pretty much intact.
Right. And I remember the particular conversation that I had with Jason, and I don't want to assign any accusations about the mushroom to him, but that pile of mushrooms that was there from the summer, he contributed this calostoma to it in probably January, and come the spring, that those calostomas were still there. Everything else was gone, rotten, taken away, but those calostomas were still looking the same. So pretty interesting.
I would like to point out that the spores of Calostoma lutescens are not warted; they are pitted, similar to a golf ball. And that's what Michael Coe says on his webpage for Calostoma lutescens, which I posted to the chat. And they looked, Chris, in your photograph, like, I thought they were reticulate. I didn't think they were worded, but the proper term is pitted. So just think of a golf ball, what it looks like, and that's very similar.
Oh, so they're like divots?
Yeah, and there's going to be a network of these dikes between the pits.
Wow.
Yeah, the edges of the pits probably look like projections.
Right.
Yep, the spores look exactly the same, and that kind of makes sense. Just looking at the link there, you were like dimples.
Dimples, yeah.
Oh, weird. You know what I'll have to do? They'll probably look cool under the. I'll have to harvest some of these spores next time I see one and
Are you
do it
going to go
under
to
the dark field.
Dark field? Yeah, that'll look cool.
Well, start with 1,000 magnification. You have to do this in oil. And
that's pretty cool.
If you add some kind of a contrasting reagent, I wonder if these are amyloid or something like that. Maybe you can get a cool presentation of them as well at that magnification.
Interesting.
Yeah, we find them in the Pine Barrens from time to time, but they're not very common. Earlier last year, I shared an observation of Calostoma cinnabarina from the Pine Barrens. So I don't have to show that observation again, but it's a common species. But, we find both in the Pine Barrens then. Who knows, maybe there are some cryptic species masquerading themselves as both of these things. They have to be sequenced. I think we're taking a lot of things for granted.
They're found, especially at Franklin Parker, I know where we find lutescens, it's found in that same spot. We'll check the spot. It's not always there. But
is it the same spot we're talking about?
Well, mostly my point is they'll show up in the same spot, right? Year after year.
Yeah, I mean, there might be multiple spots, but I remember one specific location that I think Tim Baroni came to visit us in 2017, and we took him out. FPP was in November 7th, I think, and we found them there for the first time, I think, and he dug them out and he took them with him. I'm not sure if he sequenced them or whatnot, but I just remember where the spot was.
Yeah, I think it was in Franklin Parker that I found them.
It's at Speedwell. I'm talking about a spot in Speedwell.
Speedwell. Okay, I don't remember. I remember Pine Barrens, finding them along on the side of a trail. On the side of a wide trail. There were quite a few of them actually.
Yeah, not too far away from that spot, maybe 50 or so yards down the road, I found a rare in New Jersey, Boletus patrioticus, except that it's not Boletus, it's going to be a new genus. But that thing is very rare. It's mostly southern bolete, Virginia and south to I don't know, Florida or something. But yeah, we never found anywhere else.
I've never found that species.
Yeah, it definitely, like 99.9% found below the Mason-Dixon line.
Right.
Thank you, Igor. Anything else? For anybody who's new, if you're not familiar with iNaturalist, you should get familiar with that because you can search for additional pictures that you don't have in your field guides. Beware, probably the best ones to look at are the research grade ones, because they have at least two people who have assumed that is the correct identification if you're trying to do a match by pictures. If you don't like iNaturalist, which has an app, there's also a Mushroom Observer, which you can also upload, and the people there are looking to identify things too for you. Anybody have any questions about an iNaturalist, mushroom observer, or puffballs that we can answer tonight collectively? Okay.
Not about that specifically, Sue, but since Igor, you're here, and I had a question about, and we were just discussing rarity on that bolete. Um, how common is Tricholoma fumosu luteum, which is a mushroom you identified for me here some months ago?
I would say occasional, infrequent.
Infrequent, yeah.
Yeah, but in certain years, it can probably be found in many locations. I just haven't experienced that. Didn't have that experience. It just we find that at certain foreign locations, maybe once or twice a year. I think maybe last year it was more common, or the year before, 2024. I forget, but it's not as common as equestri. Or therium. Yeah.
And also there's a chance of all of these not brightly colored yellow gilled tricholomas. The bit of confusion between the palely colored yellow gills with some sort of streaking on the cap. There's probably some confusion there, right?
Yeah. Substitutum is probably looks like it's a species group. Maybe a few entities there. I think Fumosa luteum is quite distinct. When it's fresh, I don't think you can really confuse it with anything else.
Is it more greenish?
No, it's smoky yellow.
Oh.
Deep smoky yellow color. It's not bright, but it's very rich.
Yeah,
two more tricholomas that I think are fairly uncommon. I haven't seen either of them in a while. One is called colossum or collosus. I forget. I still can't figure out Tricholoma, some of the names. There are a couple of observations from FPP Speedwell that I made and Luke Smithson made from years ago. And another one is Tricholoma grave. That one is a really interesting Tricholoma piece of work. Very unique ITS sequence that has nothing closer than 86%. And I think we've seen it a couple of times only.
And only at the one site, right? Wells Mills.
Let's see. Yeah, Wells Mills, that's an older observation. That's the one that got sequenced. And I believe I found this at FPP. I preserved that collection maybe from 2023. I haven't sequenced it. I think I tried it. I think it failed, maybe. I have to check. So yeah, those are the ones that are interesting. They're big, they're clunky, very heavy and meaty. Yeah, so the one on the right, 342, 735, that got sequenced. And Michael Quebec mentions that in their Tricholoma study, that particular voucher and sequence.
So you didn't paste the sequence in this one. You just have it at the gene bank one? Sometimes you put the sequence in here.
No, if you click on that link, it will show the sequence.
Gene bank will have it, right?
It will also say who sequenced it. I usually do that. Yeah, this is through Mycoflora fundus, and that's the sequence. You just click on that, and it gets highlighted. Double click, and the whole string gets highlighted. Or maybe you have to drag this thing. Yeah.
Yep.
Anyway, and the other, if you go back and take a look at another observation, which I think is the same thing. Yeah, that's the one I found at FPP. It was in the other one, the first one. 2023, 2 years ago. Pretty sure it's grave.
sequence,
Oh
but
wow.
yeah.
So that's pretty brown, huh? Brownish
Yeah,
red.
yeah, brownish red. I don't think this is colossum or colossus. But I don't know either species very well. I thought I knew colossum pretty well when it was more available, but I haven't seen that species since like 2014.
And the species name indicates that it's a pretty big critter, right?
Yes, yeah.
Okay.
I think it's a European species, and maybe it got sequenced. I have to check if Tricholoma colossus got sequenced. Yeah, if it's a name, if it's a noun in opposition, it's going to be colossus, regardless of what the genus gender is.
Nothing.
What? No colossus?
No colossus in INAT, unless it's because I got to choose it. No, it didn't
Let's
seem to
see.
do
I
anything.
think it's still hooked on Pennsylvania there on the left.
Ah, thank you. Okay, 65.
Yeah, I have a couple of observations. Here's Luke's. Let's see if that one got sequenced. No, it didn't. But I can
Oh,
copy.
Kira has one. Kira.
I'm going to copy a couple observations. I mean, you can do your own search on MO, but to make things simpler, here's one, and the other one I think is this one. So that's Luke's, the first link, and that's mine here, next to my BlackBerry-like phone, which I don't have anymore. I have it, but it's defunct. I like to carry it for scale because it was exactly 4 inches tall. Or 4 1/4 inches, I forget. So yeah, so maybe this is Grave, the one that you showed, Luke's. Yeah, this one I think is Colossus. Pretty sure about that one. And I remember the spot. Yeah.
So one's darker than the other, is that?
Yeah, I think it was late in the season and they got rained on, so maybe they were a little bit waterlogged. Yeah, so you can see how big they are. I mean, that phone is 4 inches at least long.
And these are all
Yeah,
sandy soils. Even the
yeah. You
Colossus
see,
is sandy soil.
they have something on there's a feature in their stipe. The bottom half, there's almost like a boot there, like a remnant of a veil, and it looks like there's a sock on the bottom half of that stipe, and there's a boundary. You can see the boundary very well in the photograph that separates the bottom half from the upper half. So I think based on that feature, you can probably identify this as Colossus. Grave may not have that, but like I said, I don't know either species very well. You have to be careful. I thought we sequenced one of them. I have to go back and see. There's probably another observation. Maybe not. No sequence there. Here's one more from FPP, Speedwell as well. A young one with a cross section. Yeah, if you scroll down, Sue, you'll see.
Oh, I was just going to take your link. She sent it.
Okay.
Well, that was awfully short.
Yeah, this is young.
So we don't have that delineation on the stipe to look at.
No, it's not kind of, underdeveloped or not apparent. Yeah, and if memory serves, this is the same spot where we find Amanita persicina every time. It's a place on one of the wider roads, and there are almost no trees in there. There are some pines and maples, but it's quite open. And this is where we found this in 2016. I'm pretty sure of the spot. I mean, I have the GPS coordinates. Obviously, you cannot see them, but I do have them. So yeah, those are quite meaty and cool tricholomas.
And we can't see them until the fall, right? Because they're all
Yeah,
tricholoma,
I
fall
think
mushrooms.
most tricholomas are fall mushrooms. I mean, you'll probably find some. I mean, the ones that grow with hardwoods, you may maybe find earlier at the end of summer, early fall. But all these conifer-associated tricholomas, they're till like late October, November, and sometimes even December. They like
Tricholomas include the the one that is the prize edible. Besides the questions,
Magnetolari. Yes,
Magalala. Yes,
the Matsutake. Yeah,
yes, Makitaki.
yeah, and we find that too. Actually, this year, Nina, John, and I went in early November to FPP, and I think we found two more locations where we haven't seen them before. It was just pure serendipity.
For those
So
who are
they're
not
kind of
familiar with,
spreading.
just to
I'm sorry,
define
go
what
ahead.
FPP stands for, is Franklin Parker Preserve.
Yeah,
We
this
have
is Franklin Parker
we've
Preserve.
been
I
doing
prefer
a
to say,
project there for 10 years or
pers-
so at least.
pardon me?
I'm just trying to describe the Franklin Parker. We've been doing it for like 10 years. Um,
No, we've been doing this since 2009. Nina and John, at least, have been doing this since 2009.
that
I
would be
think I'm
found under special projects somewhere,
right.
Jersey. What do we have? Special projects. Under New Jersey mushrooms, we have a special project since 2009. And in that particular area, there are ones that are found as typical pylons, but not in one spot. Just to clarify for newer people.
Right. It's like in the heart of the Pinelands region. So yeah, it's like the hardcore Pinelands as opposed to something like Shark River Park or um, what's the one in Clayton? I forget.
Scotland Run.
Scotland Run. Yeah.
Yeah, and I think, correct me if I'm wrong, the Burkharts have been keeping records there since before 2009, right?
Not sure, but all I know is that our species list there is probably like 700 strong.
Oh, I think it's higher than that. I just peeked at it. Oh, let me see if I can pull that up.
Well, it was like six something, approaching 700, maybe, I don't know, 2, 3 years ago. So it's possible we added a number of species to go over 700. So I can give you the exact number. John knows.
Well, we renamed them and we didn't delete the old names.
I'm sorry,
Maybe
what did
that's
you say, Sue? What about
Perhaps
names?
we renamed them and they didn't delete the old names yet, right?
Oh, I see. Okay, so there are duplicates.
Duplicates, yes.
yeah. I was just going through it, um, looking at what species of Rutiboletus are there, Igor. Um, Not Rutiboletus, Retiboletus. I'm sorry, just to see if both griseus and Venasipes are found in the proper pine barrens.
No, they're not. Griseus is absent.
Yeah, because it's not a pine associate, right?
Right, but the thing is, we have oak in there, so conceivably Griseus could grow in the pine barrens, except that probably the soil composition is wrong for it. It just doesn't
Or-
like the acidic sandy soil.
I did see there was a variation on griseus that was on the list. Are you familiar with that?
Fuscus, that name is defunct. It's just wrong. It's a misnomer.
Yep. Okay, so if
Retiboletus
it's
fuscus is a Asian species, and they just made a variety out of it for griseus in error. I think it was the Bisets, maybe.
so if I'm finding something that I'm on the fence about griseus, like is it reticulate enough, or is that reticulation a little bit too subtle? But I found it in the pine barrens in that acidic soil. It's likely Venasipes, right?
Yeah, it's Vinacipes, and it would have more of a brown gray cap or brown cap as opposed to gray cap of griseus, and the articulation is going to be more, as I call it, aerodynamic. It ain't going to be shaggy. It's going to be very smooth, polished, and more delicate. Not it will be still prominent, but not coarse. And of course, you're going to
Okay.
have the vinaceous stains at the base of this type, which griseus doesn't have.
Now, what if it doesn't have the vinaceous stains? It still kind of has to be considered vinaceous, right? Or
It's
Vinacipes.
still going to be Vinacipes. Yeah, I don't think we have griseus in the pine barrens. I don't think so. I
Okay.
mean, the only way to check would be to sequence some of the more ambiguous collections. But I bet, as Dave says, my personal limit $20 that we don't have griseus in the pine barrens.
Right. Yep.
Yeah,
Right. Thank
let's
you.
see what INAT says for.
This is griseus. Okay.
Well, click on one of them. Let's see. Yeah. How about that one in Burlington County?
Sarcastic snail put it in. All right.
No. Yeah, I think that's Vinacipes.
Right. That cap is dark.
Yeah, it's brown, and the reticulation is going to be not coarse, and
Elongated, aerodynamic, right?
it's going
Kind
to be
of.
smooth. And yeah, if you click on the other pictures just to see what the reticulation looks like on this thing. Yeah, I see those stains on the bottom.
Mhm.
Some red in there, right?
There is some red, but
Yeah.
isn't Griseus, doesn't that have kind of yellow stains at the foot?
Yeah, they griseus kind of stains all yellow when you handle this, but yeah, it has a lot of yellow on the bottom as well.
Yeah, but
The bottom line is that
the
this
ret
one is a
the
pine associate, and
yeah.
griseus
Mhm.
is a exclusively oak. Let's click on another one.
Yeah, we'll find mine if we click on enough of them. I think I still have it listed as Griseus.
You way down in the south here, Brandon, or
No,
further
I'm
north?
up around Tom's River on this one.
Is that Tom's River? No, up
Up,
here,
up,
up here.
yeah, here,
Yeah,
yeah.
I see you. No, that's not you. That's not you. That's Jason.
Yeah, I see Jason. Yeah. Oh.
Might be the same collection.
Yeah, you see, it's
Pretty.
if you compare with griseus, it's very shaggy and coarse reticulation. This one is more delicate, more refined, and again, brown cap. Yeah, I would call this Vinacipes.
Well, I
Do we
guess
have a
we'll,
section? The last photograph is sectioned mushroom. Maybe we'll see those stains on the flesh. Lots of yellow.
Now, Igor, do you remember the example of Venasipes that I showed you earlier in the or later in 2025 that stained a little blue in the foot?
Yes, and
Cool.
if you read the original description by Baroni and Ortiz Santana, and mind you, I think I mentioned this already, like probably 20 times. The type collection comes from the Dominican Republic, or whereabouts the Caribbean basin. The holotype mentions bluing in this type as well. Trouble is that once I find a bolete and I know what this is, I don't usually cut it. I try to preserve specimens at forays intact, such that people can, have more enjoyment from handling them intact as opposed to something like sliced up. But eventually we do cut some of them. Yeah. So let's look at the one in Burlington County. The one on the left near Philadelphia, or closer to Philadelphia.
Over here? That's not Burlington. That's over the river.
Yeah, this county, the one that's like a wedge. Yeah, this one. Yeah.
This one?
I thought maybe that one
That's
is. Oh, this is
Luke's.
Luke's from 2017. He says it's griseus. Yeah, that's my nice piece. So somebody should propose a nice piece name on that one.
Yeah, I'm not logged in here on iNatural, so I can't make the changes. Maybe we should plan on doing some of that on the Tolopolis next week. Go through some of our members' stuff and clean them up.
Yeah, there's
there is
tons of
something in
stuff
works
on.
that I don't know if Emily wants to talk about. Making
Yeah, there's tons of stuff both on the MO, especially
we could
INET,
do
because there are more observations there
the
that
same
just
thing.
misidentified all the time. I searched for Fistulina curtisii, and there's a lot of collections that turned out to be Suillus, probably Sabarius. I think I spoke about that last time.
Yeah, I thought you're normally on top of all of those, or you stopped doing it every single day.
I don't go to INET. Like I said, I have no presence in there, no account. You see
No, but I
this
thought you said the
situation?
same problem as MO. The MO is still not all cleaned up either.
Yeah, I slow down with MO too. There's just not enough time. I mean, I do go there occasionally, but not as often, not every day. And besides, it's off season, so
Yeah.
I mean, I don't
Well, you had a whole
propose
month
any
to clean
names
it all
for
up.
what's that?
You had a whole month to clean it all up. Whole
No,
month of December.
no, no.
Okay. I know we got other things to do at the end of the year for the club.
Yeah, for me, it's a busy period with the club.
Okay.
All the accounting and membership and stuff. So piling up. Yeah, I used to be very, very involved with MO, doing some custodian stuff like editing locations and names, things like that. But all of that is in the past. Now
You
they
ready to call
even
it a
said that.
night?
Never mind.
We're ready to call it a night, or anything else? Somebody wants to bring something else up?
So
I'm
you
ready.
said Telopolis?
Galapagos next week, yes. Jason is doing the presentation.
Jason's doing presentation.
Yes,
Okay.
unless you didn't volunteer, right?
No, I didn't volunteer. No.
Yeah.
I will get bogged down by this, and it will take me forever to.
I mean, we still have another subject, Amanita to sections. You could volunteer for that. That's on the list of potentials sometime over the next couple months.
Yeah, I have that old presentation that's probably will take me at least an hour to present.
Yeah. Okay.
Yeah, Telopolis. One of the collections got sequenced from Thompson. And I think we find that Telopolis species there all the time. I think it maybe comes from Helmeta because but I'm not sure. So I got it sequenced, and apparently I already had sequenced that species before, but from a totally different habitat and location. And the name that sort of transpired, probably incorrectly, is Appalachiensis. I think its name that was proposed by Yves Moreau for his Canadian collection, and my sequence from Washington Crossing from a number of years ago matched that, and that's why I called it Appalachiensis. But most likely it's a misnomer. It's a misidentification. But Tulopolis is a very interesting genus. Lots of boring brown mushrooms, but that's where the interest comes in, because apparently we have many undescribed species, and the ITS is an excellent barcode to separate them.
And ITS is being the portion of the DNA that helps
ITS is the
identify two species and genus.
internal transcribed spacer.
Well, that's nice that it works in that genus. Wow.
So let me show this observation, just kind of a sneak preview. Let me find the observation number, or maybe here, because it's in my lists, because it was sequenced, so I can find it quicker. I'm going to post the link in the chat so you can open it up, Sue. There it is. So it had a very unpleasant smell and taste. It was disgusting. But you can see it's a fairly, I wouldn't call it large, but medium-sized. It's readily stains brown everywhere. And the collection was not in a particularly great shape, but it likes to grow in clusters. This is one thing about the species. It likes to grow cesspitose. This is one thing that I noticed, which is a repeating theme for the species. So this collection and the one from Washington Crossing that I reference here in the notes, or maybe actually no, that one is from also from Helmeta. But I probably do discuss the Washington Crossing collection there as well. Yeah, you can click on that name, and maybe it will show.
Which name? Where you wanted me to click on?
Well, if you scroll down.
Scroll down.
And click on the on this name, Appalachiensis, in the proposed names, it's going to give you all the observations that have that name, and you can probably find the other collection. Click on the this name in the about this taxon on the right. Yep.
Oh, yeah, okay. Sorry, wrong one. This
the top one.
Name, right? Or do you want
this name? Yeah.
That one, okay.
Is it doing anything? Oh, okay.
Yes,
and
it says drawing these pictures.
Yeah, and apparently there's probably multiple things in there that are not Appalachiensis that are all different. If you scroll down, yeah, mine is on the right. The first line. Yeah, this one. So this one came from Washington Crossing, and unfortunately, all the trees there got cut down because they're building that visitor center, and that place is decimated. So it was bitter, and you can see it's growing in the cesspitose cluster. Same ITS as the one from Thompson. Not as brown, more pretty, but same DNA. And
well,
my
we're gonna
usual
save it for
fashion.
next week.
Yeah. But like I said, this may not be the correct name, and maybe this is something totally undescribed. And we'll never know until we start sequencing type specimens for all these obscure species.
Do we always have the type specimen available to take DMA from? Or
Well, It's in a herbarium. The question is, how old is it, and whether the herbarium, like say New York State in Albany, will allow you to actually sample something. You have to be like a member of the ivory tower of mycology.
Yes, I know we can't. I'm just wondering if you don't no longer have the original specimen, does it no longer get to be called the type specimen? I mean, if somebody loses it, does that mean it's gone, or
Yes,
something
if the
else?
type specimen is gone, they either create a neotype, or maybe there is a syntype or isotype floating around somewhere, because sometimes the collection is split. So the type goes to one facility, and another portion of that specimen will go to another herbarium, and that's going to be called an isotype. And there's also such a thing as syntype, which is a different collection that was used to create a species concept. So maybe another location, but the description of the species is also based on that collection as well. And this is where mistakes sometimes are being made, because we don't know to certainty if both collections are the same species. So that's a syntype. So a neotype is created when the original specimen is lost.
Okay.
And there's also another type of type called electrotype. And electrotype is created when no holotype is designated. So if, say, Peck described a collection of a bolete, but he failed, and he used a particular collection, right? And that's, that went to the herbarium, but he failed to designate that as the holotype. Somebody else may examine it and say, I'm going to designate it as a holotype, but in this case, it's going to be called a lectotype, because the original person who described it did not designate that. Okay, so it has to be called a different kind of a type. So a lectotype is the correct name for that collection from that point on, I think.
Thank you. I'm sure we'll forget this by tomorrow.
No, yeah, I mean, it takes a while to sort of, you have to sit down and memorize this, or
Yes.
try to logically remember, and then it's going to make sense. But it's not something that goes in easily into your head.
Yes.
I mean,
Because I'm not going to participate. I'm not going to create holotypes, I'm sure.
Yeah, you just have to remember that holotype is the original specimen from which the
Yes.
description was taken. And a portion of that is a syntype. Sorry, isotype. And syntype is a different collection from which the description was also drawn for that species. So it's like an additional material that is not a holotype, but a participant in the description, I think. Yeah, it gets kind of confusing. So, yeah, the Appalachiensis, I think it could be a singer species. And if that's the case, it probably dates back to the 40s or 50s, maybe. I have to look it up. Index Fungorum to the rescue. Singer, 1948. And probably in Florida. You know what? The holotype is from North Carolina by Coker. And the taxonomy nomenclature here is not very straightforward, but that's another story. So again, we may have a problem when somebody renames something, because Coker published this as Phaleus variety minor, but that was an invalid name for some reason. And then Singer came over and called this Appalachiensis. Interesting, right?
Yes.
So when you do all this research, this is one of the most important things, to be able to work with literature and interpret it correctly. It's probably one of the most difficult parts of trying to publish something is to make sure that it hasn't been published before you. So this is where that skill of being able to work with the literature, find specimens, comes in handy.
We have Chat GPT now, so I think somebody else is going to do the work for them.
Are you being facetious?
No,
No?
because it was just an article about somebody basically writing a TV show and they had pictures of their screens and they had Chat GPT tabs open, and they were saying, oh, they're describing the next episode, letting Chat GPT decide the path of the episodes.
Oh,
Anyway.
God help us.
Yes. Well, for creative forces, that makes sense. For literature, I don't know. You have to trust the author too. There's some authors I may not trust as much as others. You know, just because it's written down does not make it correct.
Yeah. Well, for mycology or biology, where we deal with physical specimens and real descriptions, yeah, I mean, ChatGPT, I don't think so. And you have to do your own research. And you have to come to the conclusion that what you read and see or handle is the real McCoy, and there's nothing beyond that. You have to convince yourself of that. And this is not a task for some AI. There's never going to be a substitute for this. I hope not.
Well, that we'll call it tonight, the end of the session. I hope everybody comes back next week and the topic of Telopolis, which is in the bleat family. And, isn't it? So good night, everyone.
Anybody wants to stay around? Not many people left.